So, we have finished chapter 3. We will move on to chapter 4. We've finished chapter 3, which covers macromolecules, DNA or nucleic acids, proteins, carbohydrates, and lipids. Okay? The next step up in our progress of biological organization from atoms to molecules to macromolecules next is organelles. Right? And really we talk about functional parts of the cell. Okay, so really we're going to talk about parts of the cell. That's what chapter 4 is. Okay? But first let's compare different types of cells. All right? First off, what you see up there, the fundamental units of life. So these 4 bullet points compose what we call the cell theory. Okay? The cell theory. The parts of it is that, number 1, all living things are made up of cells. If it's living, it's made up of a cell. Otherwise, it's not living. So desks, not living, not composed of cells. Okay? The next one is the smallest unit or the simplest collection of matter that can be considered of living. So the smallest living thing is a cell. All right, so the smallest thing that can be living is one singular cell, a unicellular organism. Okay? So if all living things are composed of cells and the smallest living thing is a cell, where did cells come from? Great question. Where do cells come from? Well, that's the next part. Or all cells are related by their descent from earlier cells. Another way to say that is all cells come from other cells or pre-existing cells. Okay? Those 3 are really the basics of the cell theory. Okay, and what do we talk about when we say the cell theory? What's a theory? There's a theory of evolution, there's a cell theory. What's a theory?
It's kind of like a hypothesis that can't be proved.
Can't be proven,
Yes.
can't be disproved yet. Right? Because we can never, so even when we talk about hypotheses, right, we can never really prove, beyond a shadow of a doubt, prove a hypothesis correct. We can prove a hypothesis to not be true, right, by experimentation. That's what scientists do. Okay? So a theory is a collection of ideas or statements or conclusions that have stood the test of time. Okay? We have not had a point where any of these 3 things, that the smallest unit of life is a cell, that all living things are composed of cells, and that all cells come from pre-existing cells, those 3 things have not been disproven. That's not to say it couldn't be at some point. Maybe we don't know yet. That's why it's called a theory and not a law. Okay? Difference. Evolution is a theory. We hold it as truth. Okay? It's been proven time and time again. Numerous types of, numerous ways of proving that evolution is there, but it's never been disproven. Okay? So even though evolution is a theory as well. All right? How do we study cells? And we're going to go through these really quickly. Okay? Oh, excuse me, sorry. All right. Next couple slides we're going to go through real quick. What did you use in lab to study cells? Microscope. Okay? Why do we use microscopes to study cells? Because we can't see them with your naked eye. All right? Most of them are too small to see with the naked eye. What's the largest single cell thing? Many of you probably had couple single cells for breakfast this morning. Eggs. All right, the largest single cell is an ostrich egg. Okay, that's all. It's a single cell. Okay. About the size, frog eggs. Okay, frog eggs are about the limit what you can see with the naked eye. Okay, and they're single cells. So you can see some single cells with the naked eye, depending upon what they are. Most of them, you need the microscope. Okay, so most of the time, and what you'll be using, you're familiar with, is light microscopy. Okay, light. In lab, you use the light microscope. Okay, light microscope uses some type of illumination, whether it be reflection of sunlight or ambient light, or some type of light source to shine light through whatever specimen you're looking at and allows it to you to view the image. Okay? The lenses, the ocular lens and the objective lens, they bend the light so that the image, once it ends up in your retina, in your eye, is magnified. Okay? And for the most part, light microscopes are great. Okay? We can use them to visualize single cells, right? Most of the time, they're pretty large, eukaryotic large cells. All right, so we talk about this in lab, magnification, resolution, contrast, what they are. Magnification is how big that image is presented, right? It's a ratio of an object image to its real size, right? So if you end up with a 400x total magnification, that means the image is 400 times bigger than its actual size. Resolution, okay, is how well or how clear an image is. And then contrast is visual differences in parts of the sample. You want to have high contrast so that you can see surrounding light versus what you're actually looking at. All right, so those are three important parts or parameters of microscopy. Now, the light microscope has been around for 150 years or so, about mid-1860s, when microscopes were first invented. Okay? It's come a long way, and we do lots of funny things with it now. Okay? With light microscopy. Well, here's the order of cells stuff, right? Human organisms, and basically this is about the frog egg is about the size of a millimeter in diameter. You can basically see them at about. Then light microscopy can be used to see most plant and animal cells. You can kind of, towards the higher magnification, you can kind of see single bacteria, okay? But then from that point, you have to go to higher resolution. This super resolution microscopy, that's higher advanced stuff that we're not going to be using, right? More higher technology in terms of being able to see smaller and smaller things, all right? An electron microscopy we can use to see very small parts of the cell, all right? So what you can see then is you can basically see the whole cell, and you can start to make out, depending on how good of a microscope you're looking at, you can start to make out parts of cells. Start to, okay? Organelles. You can see the nucleus, you can see the nucleolus, right? You can observe the plasma membrane depending upon the staining you're using, and so on, all right? But they're hard to really see clearly. So the next step is to utilize electron microscopy, which increases magnification with better resolution as well. What the hell was that? All right, so scanning and transmission electron microscopes what they do, instead of light, they actually blast electrons across a specimen. And those electrons are either absorbed or they are filtered through, right, which leaves an image behind, okay? Scanning electron, scanning means they actually look on the outside of stuff, okay? A transmission, the electrons go through and you can actually see through the internal structure. So a scanning electron microscope, it gives you a three-dimensional image of what the surface of something looks like the surface of a cell, surface of a fruit fly, whatever it might be. Transmission, you can actually see parts and very distinct structures within cells. All right, so this is what it looks like. Okay, so here's light microscopes. You can use a bright field. You guys did this, right? You looked at bright field images of your epithelial cells and you had them stained with some blue dye. This is phase contrast, so it looks a little bit different. All that is just bending the light a little bit so you can see the differences of how the cells are bending light, right? Nemarsky is another difference of phase. But here, what you end up with in this one right here, in fluorescence and confocal, it's still light microscopy. Why does it look so different? Well, what you're doing is you're actually exciting something, a chemical or antibody, a dye, with a very particular, a specific wavelength of light. And with that wavelength of light, that dye fluoresces and gives off another wavelength of light. Okay? And then you take a picture of it, and it looks green and red and blue and all that funky stuff. All right? But it's a technological advancement of light microscopy. And then there's confocal, which is the same thing. And then you get your scanning and transmission electron micrographs, where the scanning shows the outside of stuff, while the transmission actually sees the internal structures. Questions on microscopy? Basics of it. No? Yes? Maybe? Are you awake? I don't care. All right, so the recent advances. We can also talk about cell fractionation. Biochemically, we can separate parts of cells. Okay? Nucleus is a very dense structure. Okay? It has lots of DNA, it has proteins, it has stuff in it. So you can utilize put a tissue in a detergent and spin it down in a centrifuge, and what will end up happening is parts of it, of the cell, will go down to the bottom because it's heavier than other parts of the cell. Right? So I could look at nuclear proteins versus cytoplasmic proteins just by the fact that a nucleus is more dense than the rest of it. Okay, that's what cell fractionation is. All right, so biochemically we can separate parts. We can separate the nucleus from the plasma membrane. We can separate parts of the mitochondria from the rest of the cell. Okay? So, what do we know about cells? They come in two types, don't they? What are those types? Can't understand when you're mumbling. What are they? Prokaryotic and eukaryotic. All right, that's better. So prokaryotic and let's move this over here. Okay, eukaryotes. What's the difference? So prokaryotes are bacteria. Okay, yes. What else? What's the big difference? Eukaryotes have membrane-bound organelles. That's organelles, okay? Prokaryotes do not have membrane-bound organelles. So the nucleus that's in a eukaryote, the mitochondria, the Golgi apparatus, the chloroplasts, whatever other parts of the cell that you want to put into the eukaryote part are membrane-bound. Okay? They have membranes around them, parts of the plasma membrane. Okay? Prokaryotes do not have membrane-bound organelles. Let's talk about what both of these do have. What do they have? What do all cells share? They have some type of DNA. Right? Some type of genetic material. What else? A plasma membrane. They have to share, they have to maintain some type of inside-outside part, a plasma membrane. Okay? Anything else? No? Yes? Maybe? They have to have an in part, right? So they have some type of cellular cytoplasm. Some type of watery fluid that's inside the plasma membrane. One more thing that all cells have. All cells have.
Mitochondria?
No. Because bacteria don't have mitochondria. What else? Any other guesses? Ideas? Who said it? Ribosomes. What do ribosomes do? Ribosomes do what? Jump up and down? No, they don't. What do? They are the site of protein synthesis. Okay? So, all cells have to have some type of genetic material, DNA. They have a plasma membrane, which separates outside from inside. They have cytoplasm, which is inside the cell. Okay? And they have ribosomes. Okay? So, bacteria and archaea are prokaryotes. They do not have membrane-bound organelles, while protists, fungi, animals, plants all consist of your eukaryotic cells. So we have two types of cells, eukaryotes. There you go. Basic features of all cells. Okay? They all have these things. So chromosomes, you can say, is DNA. Plasma membrane, we wrote. Cytoplasm is cytosol. Like they are basically synonymous. And ribosomes that make proteins. So all cells, whether they are bacteria, prokaryotic, eukaryotic, protists, fungi, you name it, they have to have those four things. Understood? Are they the same in bacteria and or prokaryotes and eukaryotes? No, they're not the same. Ribosomes may differ. Ribosomes that are found in bacteria may not be the same as you have in eukaryotic cells. They will be different, but they still do the same thing. They still make proteins. The structure of ribosomes in bacteria is different than in eukaryotic cells, but they still do the same thing. They still make proteins. Understood? Plasma membranes will be different, but they still are the boundary from inside and outside. All right? So, prokaryotic cells have no nucleus. Their DNA is not separated from the cytosol. It is collected in one area called the nucleoid. And again, this is your big one, no membrane-bound organelles. And they do have cytoplasm that's bound by this plasma membrane. Yes, sir.
Is membrane-bound organelles, is that like proteins on a membrane or something?
It's not proteins on the membrane. You'll see that there is a very complex membrane system that's within animals, eukaryotic cells. Okay, so if they have so here's a plasma membrane of a eukaryotic cell, right? The nucleus, okay, is there. But if I look at this part of the nucleus, you'll see that there's actually a membrane that's separating inside the nucleus, right, in versus out. There's a double membrane that's around that. And there's a double membrane that's around a chloroplast. And there's a double membrane that's around a mitochondria. All right, so they have this separation. It's all part of that whole system. Okay?
Making it more complex, yeah.
It's extremely more complex. All right, but it's not like just proteins that are covering. There's parts of the plasma membrane that's throughout every organelle. So it's compartmentalized and separated. All right, so here's a picture of E. coli, right? A nice prokaryotic cell, right? Diagram on the left and transmission electron micrograph on the right. All right, what you see here, this dense region on the inside, right here, right? When I say it's more dense, when you're looking at a transmission electron micrograph, the darker it is, the darker areas, that's more density, more stuff is there. So that region on the inside is the nucleoid. It's where the DNA and chromosomes are packed together. Okay, this is a diagram showing this. The little fuzzy stuff around the outside, that's called fimbriae. It allows for E. coli to attach to each other, right? But there's no compartmentalization. The DNA is not separate from where the ribosomes are. It's not separate from where all the other stuff is. Okay? It's floating around in the cytosol. All right? Clear on that? Let's compare that to some eukaryotic cells. Eukaryotic cells have DNA that is compartmentalized. DNA is not found, normally not found, outside of the nucleus in eukaryotic cells. Okay? We have membrane-bound organelles, and cytoplasm is still there. So we have this stuff in common, right? Cytoplasm between the nuclear membrane, nuclear envelope, and the plasma membrane. All right, and in general, eukaryotic cells are larger in size. All right, so some differences between the two. You guys, man, dropping stuff, going to the bathroom like crazy today. Okay, so questions on basic eukaryotic cells. We'll see a picture of them in a second. All right, but first I want to talk about the plasma membrane. Some things that we're going to have in common. Okay, today we're going to talk about the membrane and the ribosome a little bit. Okay. I'm not going to go over all the different organelles that are present in eukaryotic cells. On Friday, I'm going to provide you a list. And as a group, actually, you already have the list. It's already on your study guide for exam 2. And in groups, you're going to go through and write up and compose a study guide, okay, for those exams, for those organelles. Understood? So we're not going to cover them because I could just rattle them off. And I have rattled them off in past classes, so the lecture videos are up. You can actually have me talk about it to you and teach you about it. But you're going to fill out that study guide. That's going to be your class assignment. All right? But the plasma membrane has a distinct structure. It is a selectively permeable barrier, all right, that allows certain things in and out of the cell while excluding certain things from entering or exiting the cell. The general structure of a biological membrane is a double layer of phospholipids. Okay? So, what does that look like? What's a phospholipid look like? Two fatty acid chains in a phosphate group. So there's your phosphate group, and there's your fatty acid tails. Okay? So I'll label that. This up here is your phosphate. Over here is your fatty acid tails. Okay? Phospholipid, it's a double layer. So how is that layer, here's one layer, right? Looks like my daughter's drawing stick figures, right? So there's one layer. How's the other layer going to be arranged? Inverted, right? So that the tails are pointed towards the tails, right? Okay, so it's going to look like this. Here's your double layer, your second layer. And what that gives you is what? What do we know about the phosphate parts of these phospholipids? They are what? They're polar and what? Polar and hydrophilic. Polar means it's what? Plus or minus, right? They're charged. Yes? Okay. So plus or minus, they're charged. What else is charged? What molecule do we talk about extensively that has a charge to it? Water. So these charged heads, these polar heads, like water, they are that is a Y, hydrophilic. Right? And down here, I actually probably should have put that in blue. This area right here is nonpolar, where the fatty acid is, nonpolar and hydrophobic. Okay? That's the basic structure of cell membrane, of the plasma membrane. Okay? Basic structure. Now there are a lot more complexity into that. There's proteins, there's carbohydrates that do certain jobs that are part of this, but this is the basic structure. And the hydrophobic and hydrophilic regions, that gives the membrane its selective permeability. Because things that are too big can't pass through. Okay? Things that are charged can't pass through. Okay? Because of the difference of it. So here's what it looks like, though. Okay? You have proteins that are embedded in this. Here, right? You have proteins that are embedded that actually could be on the side, attached, or they span the membrane. Right? In this case, this one right here is a channel. See, it has a tunnel that goes through that allows things to go through that couldn't pass through the membrane itself. Couldn't pass through the phospholipid bilayer by itself. Maybe it's an ion. Maybe it's a molecule that's too big to pass through by itself. Okay? So the proteins are embedded in there. Okay? And they have this, there's lots of different parts to a plasma membrane. And this is what we call the mosaic model. What's a mosaic? Okay, in terms of art, it's a picture that's made of what? Bunch of little pieces. Bunch of different parts. And that's what a mosaic is. This is a mosaic. It has a bunch of different parts. It has phospholipids, it has carbohydrates, it has proteins, different types of proteins, right? It has carbohydrates that are linked to lipids. Yes, Brett?
You start sticking stuff into that, how does it not destabilize?
So the stuff that's stuck in here is arranged in a certain way. The way this protein is made in the endoplasmic reticulum, it's actually embedded into a chunk of membrane. So then it's surrounded by phospholipids and then stuck in there. The protein itself has parts of it that are nonpolar. Okay? So nonpolar amino acids that make up the part of the protein that spans the nonpolar region. Okay? So it's not going to disrupt that because it's structured a certain way. Okay? And then it's embedded in part of the membrane and then moved out there. Okay? The other part of this model is that it is not a stagnant, stationary thing. These proteins are embedded in here, but they don't necessarily stay there. They get moved out, they get moved around, they can move it throughout the entire membrane. Okay? So not only is it a mosaic, it is a fluid mosaic. Fluid, meaning it is dynamic, it can change. Okay? Parts of it can change. When you get up into more advanced classes, cell physiology, right? You take a cell biology course, you'll talk a lot about these proteins and how they're made and how they're structured and how they're embedded into membranes. We'll talk about how the membranes can flip one in and out. We call a flipase and a flopase. You can actually flip the inside phospholipids so that they're on the outside, and so the outside are on the inside. Flip them around. Okay? That's all like cellular physiology, how cells work to do things. Okay? But understanding the structure is important. Okay? One other thing about cells is that they have, we went through the size limits, right? We said that basically the biggest single cell you have is an ostrich egg, and from there, you get a fertilized egg. What happens? Does a chicken egg get any bigger? No. Right? It's laid, it's fertilized, and then what the chick starts to develop inside of that, right? And so you end up with an egg cell that then ends up becoming a chicken, a little chick that hatches out and that has thousands of cells. Did the volume get any bigger? The ultimate volume of the egg, did it get any bigger? No. Okay? And so we have this, what's called an upper limit to size of cells. They can only be so big because of their surface to volume ratio. Their surface area to volume ratio. What's the surface area? What's surface area? The space that's on the outside. So I've had a basketball, right, up here, a nice sphere. Right, and I'm not going to try to draw a sphere in 2 dimensions, but it's a circle, right? But a sphere is kind of what most cells are structured as, right? You think about an oval sphere as an egg. The shell is the surface area, right? The volume is what's inside of that surface area. And then there's a certain ratio. The smaller cells are, the higher that ratio is. The bigger cells are, that ratio distorts. Okay? So you see here. Okay? So if I have this, say this is your egg right here. Right? It's pretty big. It's, a couple inches in length, about an inch wide. Okay? Its surface area, we'll say, is, in this case, the egg is 150. What's inside of it is about 125. Its surface area to volume ratio is close to 1, about 1.2. okay? Given the mathematics you have up there. Think about an egg. Intuitively, think about an egg, okay? Now, that egg is a single cell, okay? Its surface area to volume ratio is pretty low, about 1, right? And now that egg's fertilized, I said, did the volume change? Yes or no? No. But inside of that single cell, to start as a single cell, it begins dividing. Now it goes from 1 to 2 to 4, 8, and there's a whole bunch of developmental biology that goes on there, and it's really like some cells are growing and other cells aren't, and whatever it might be. We're not going to get into that. That's a developmental biology class. However, what you end up with is a chick that has thousands of cells, and you have not increased surface area or volume. You have not increased the volume of that, but the surface area inside has changed dramatically. All those cells have divided, right? And now just the plasma membrane is there. So now you end up with 750 or however many cells you end up with. Volume doesn't change, right? So your surface area to volume ratio goes sky high, okay? And that is important for metabolic rate, all right? The ability to move things across the membrane. You have to have a high surface area, all right? If that doesn't happen, movement of things, diffusion stops or slows down dramatically. Okay, so surface area to volume ratio is important and gives a size limit, an upper limit of size for the size of cells. Okay, so this would be our next 2 slides, and the next would be our last slides. What do we know about eukaryotic cells? Right, we know there's a plasma membrane. We know that there are parts of it, they're membrane-bound organelles. You're going to go over the nucleus, right? We're going to go over ribosomes and Golgi apparatus and mitochondria and lysosomes and microtubules, parts of the cytoskeleton, okay? And flagella, and what they are, right? Those are all parts of an animal cell that do certain things. Most animal cells have these things. Not all animal cells have flagella or cilia, okay? Everything else up there, for the most part, animal cells have. They have plant cells, much different. They have a central vacuole. They have chloroplasts, right? They have cell walls. They still have cell membranes within that cell wall, okay? And they have junctions between cells, right? They have openings through the cell wall that allows cells to communicate with one another, right? Much more complex than your prokaryotic cells. They still have a nuclei, they still have ribosomes, they still have they still have Golgi apparatus, right? They still have microtubules. Okay? Questions on structure of plant and animal cells. Basics. All right, so here's some examples of human cells from the lining of the uterus with a colorized transmission electron micrograph. So it's colored, so you see the structure of the membrane, you see the color of the DNA, which is in purple, right? You see cilia, that's up here, right? And the nucleus and the nucleolus. You can kind of see these darker regions right there are parts of the endoplasmic reticulum. Okay, here's a scanning electron micrograph of yeast budding. All right, and then you have a transmission of a micrograph of a single yeast cell showing your nucleus and different parts, vacuoles, etc. Okay. Then you have the same for plant cells. All right, a chlamydomonas. All right, scanning electron micrograph, show you a flagella. You have the chloroplast, they have a cell wall. Here's cells from duckweed. You have your lots of chloroplasts, nucleus, nucleolus, central vacuoles, okay, etc. Questions on plant versus animal cell? Okay?